Thanks everybody for coming out on a frigid evening for a presentation about filter feeders around New York Harbor, specifically Newtown Creek. My name is Will Elkins. I'm the Executive Director of Newtown Creek Alliance, and joined by colleagues from Newtown Creek Alliance and Billy and Oyster. Let me just stand up and introduce yourselves real quick.
I'm Gus Berry. I'm the Restoration Associate here at Newtown Creek Alliance.
I'm cut you off. I do environmental education at Newtown Creek Alliance.
Tanisha Swift, Associate Director of Engagement Project with Big Lakes Project.
Hi everyone, I'm Illusion Rivera. I'm the Education Outreach Manager at the Billy and Oyster Project.
Hi everyone, I'm Jada Henry. I'm a Restoration Field Technician at the Billy and Oyster Project.
And we're also joined by a lovely partner of ours, artist Priscilla Stadler.
Priscilla. Hi everybody.
Yeah, so we're really excited to have you all tonight, and we're going to go through some presentations and really have a conversation about some of the stuff that we're working on as organizations and in general, and then we have a interactive, fun, hands-on activity afterwards as well. So a little bit of background for those who don't know, just to situate where we are. We're focused on Newtown Creek tonight. Newtown Creek's a tributary of the East River. It's about 4 miles from where we are here at Hunters Point to the furthest reaches back in the English Kill section. It's the border between Brooklyn and Queens. It's part of New York Harbor, so it's a tidal waterway, and it's brackish water, right? It's water coming in from the ocean, mixing in with fresh water coming down from the Hudson, and it's an amazing place, and it has a lot of history. Not so great all the time in terms of the contamination that's here and the environmental challenges that are posed. It's an industrial waterway, so aside from the new residential happenings here at the mouth, the majority of the creek is zoned for heavy industrial uses, and it's still a very active waterway with a lot of shipping, tugs and barges coming in and out, transporting materials. So that sometimes presents a challenge, right, for when you're thinking about ecological restoration and how to accomplish some of those goals. Newtown Creek Alliance is an organization. We've been around since 2002, dedicated to restoring, revealing, revitalizing the waterway. Those may be large generic words, but essentially we do a lot of advocacy work, working with agencies like the EPA and the state DEC on historic contamination, on water quality issues. We do a lot of education, including in this space. We run an educational program here and our headquarters at Kingsland Wildflowers. And we also do a lot of hands-on work, as demonstrated in this photo. So many sites where we're doing landscaping, trash cleanups, etc. A lot of volunteer events, and there's more to say about that. Our partners, Newtown Creek issues. So to summarize one of the more contaminated waterways in the country, there's a handful of issues that we're really concerned about. One is combined sewer overflow. Show of hands if you've heard of combined sewer overflow before. Love a good, well-informed audience, right? This is the sewer system that we have in older cities like New York, where storm drains on the street are connected to the same drains we flush our toilets into, and they go to a treatment plant, but when it rains, the system gets overwhelmed and it goes out into the waterways. So we have on Newtown Creek over 1 billion gallons of untreated sewage every year that's discharged to this waterway. It's a problem throughout all of New York City. There's over 20 billion gallons throughout New York on an average year. Newtown Creek is an example of a place where the problem is more severe because of a few factors. One is that the waterway is very narrow, right? So it's not a big water body in terms of volume. So you think about it like a bathtub, like it's a small bathtub, and if you open a pipe and you're dumping sewage into it, it's going to quickly fill entirely with sewage. Whereas a place like the East River or the Hudson or the Upper Bay out near the Verrazano, those are much larger bodies of water, so you put the same amount of volume, it has less impact. The other concern is that it's a very stagnant body of water, so it doesn't move that much. East River and Hudson have very fast currents, so stuff can wash out more quickly. But here, when sewage flushes in, it really sits there and festers for a while. So this is further back in the creek, and all this is right near one of the large outfalls, so it's pretty nasty stuff. Lack of access is a big issue. We don't need to focus too much on, but essentially the creek has very little places where you can get to it. There's great little parks like this here at the mouth, but the vast majority of the creek is inaccessible. So that has really helped perpetuate states of neglect and abuse because no one can see it, no one's fighting for it as much as if they were engaged. Toxic sediment. The creek was, one of these birthplaces of the Industrial Revolution, starting in the 1830s, 1840s, and so we had a lot of nasty companies that were dumping all sorts of stuff in the water, and a lot of that is settled down into the bottom of the creek. So this mud that you see here, highly contaminated with all sorts of carcinogenic chemicals. That's why it's a federal Superfund site and why the EPA is here to clean it up. So don't go out and touch the muck is a takeaway. Don't go touch the water, especially after it's rained further back in the creek. And then obviously the other big issue is ecosystem needs, and that's why one of the big issues that we're tackling with Billy and Oyster and why we're here tonight. Billy and Oyster Project, Tanisha, you want to say a few words?
Actually, I'm gonna
Oh, okay, Jay, that's right, yeah.
Yeah, so welcome everyone. If you haven't been informed before, Billy and Oyster Project is a not-for-profit here in New York City. Our headquarters are at Governor's Island, and our goal is to engage with 1 million students, particularly, to have their participation to help us get to 1 billion oysters by the year 2030. So why bivalves, why oysters? So we like to focus on 3 main reasons why our species of choice is oysters. The first reason being that they are filter feeders. So there's that natural occurrence of them. Doing that actually helps us clean and clear our water bodies and our water columns. So they're really good at doing that. The second thing that they do for us and for the ecosystem is as they're creating their reefs, as they're going through their life cycle, growing on each other, creating their large reefs, they provide biodiversity and shelter and habitat for other living organisms. So we have filtering the water, we have building habitats for other biodiverse species who come to our waters or come back to our waters, and then also the creation of these really big oyster reefs, similar to how they were in the past, can create essentially like a natural barrier for our shorelines. So thinking about climate change and how the weather here is a lot different than it was in the past. Even thinking about the storm surges that we get during the storm seasons, that can really help us with the wave action that we have been getting on our shorelines. I don't know if you want to talk about history?
Well, a little bit of the history is that, both throughout New York Harbor and also Newtown Creek, this was an amazingly rich ecosystem for thousands of years, and the indigenous people, the Lenape, utilized that. You ate the oysters, ate the clams, all the fish that were here, and with the transformation of the area, again, beginning really in the 1800s with industrial uses and a growing population, a lot of those ecosystems were totally decimated. There's a lot of great documentation about the history of oysters in New York Harbor, and the vast quantity was here and how quickly they were decimated, and then also how the pollution later on made them unsafe to eat. And that was basically 100 years ago. The city said it's not safe to eat oysters. People are literally dying from that, and so it has not been safe to eat oysters from New York Harbor since then. But that also goes the same way with the creek, is that we had a lot of other, this is basically what Jada spoke to, water quality, habitat, but we had a lot of our ecosystems destroyed. And the filtering feeding is obviously very important, both in terms of like a natural function, when we think about New York City before it was a major metropolis, but also now where you have all these influxes, right? We talked about all that sewage going in, so it's not just pathogens, but you have nitrogen and all these additional pollutants coming into the waterway. So the role of things like oysters and mussels are even, you could say, are much more critical now because the impacts are so much more severe as well. So let's talk about oysters. We talked about how much damage has been done around New York Harbor and specifically Newtown Creek. But despite all this contamination, In Newtown Creek, we're talking 170 years of dumping of oil and chemicals and et cetera. But a lot of this wildlife is coming back. So we did a wild oyster survey a few years ago, going out at low tides because they're in the tidal column and usually down very low. So you really got to find the lowest tides to go out there. And we looked at all the shorelines to see where are oysters growing already. And some pretty sizable ones there, as you can see, that are there. So we documented about 1,000 throughout Newtown Creek, and about 150 in one of the tributaries that we'll talk about in a second, called Whale Creek. So these are just some of the photos from the survey. Sometimes the shorelines are very hard to get to. We had to sort of go underneath this deck here. This place had a lot. You can see them sort of sticking out on some of these metal structures, concrete wall. Here's some folks helping with the survey. We partnered with the North Brooklyn Community Boathouse on some of this. Sometimes you need a small boat to get in there. And this is a little map of the oysters that we found. I guess the blue is just the Queen side of the creek, and the red is the Brooklyn. And so these are basically numbers, which it's like a property. A property shoreline might be 100 feet or it might be 500 feet, little clusters that we're counting. So you can sort of go through, and again, these aren't huge numbers. 6 oysters on the property next door is not a huge amount. Although I feel like this morning
It's gone up because I was out there today we had a really big low tide today, and I think I counted at least a dozen. So it's increased.
So it is changing, and obviously some of these, we found one oyster on a whole shoreline back here. Not that impressive, but the other thing that's kind of interesting just to talk about real quick with this map is that this is a truncated version of the creek. So we didn't really see any oysters beyond here. The other thing I'd mention is about 10 years ago, we also did an oyster survey, and in that, I didn't see any oysters basically beyond this area. So it's actually kind of cool to see, in the past 10 years, there's more oysters further back into the creek. And the reason why there would be more oysters at the mouth is that, one, you have all this amazing restoration that Dillian Oyster is doing throughout the East River, so there's more spats or baby oysters floating around coming in through here. And then, hopefully the water quality's been improving as well, so they can seed further back in. But we're gonna talk about Whale Creek, this little tributary of Newtown Creek, is the focus of our site. And one of the interesting thing is that you see this is the largest cluster of oysters that we saw, which is one of the reasons that we selected it as a site. So I'm going to pass it back to Jada and Illusion to talk more about the Whale Creek installation.
Thank you. So for the Whale Creek location, just a larger or more zoomed in map to tell you where the installation took place that we're going to be talking about. So we had 2 different structures that were placed in this installation. The date isn't here, but it was August 22nd, 2024. And so this is a modified oyster research station, and this was used within, where I showed you on Whale Creek, and it just has really cool visuals of how they went about creating it and modifying it from what we usually use it for. The second structure is the e-concrete disc, or the eco-disc. A really cool structure as well. And here's an example of it being used and placed within Whale Creek. And there are also photos that were added of the 30 e-concrete discs that were manufactured actually by volunteers at Newtown Creek Alliance. So as you can see, we have the molds here, and some of the work in action, and then of course the product. So 20 modified ORS cages were also assembled. It's a different kind of structural process. It's more of getting the materials and assembling it versus creating the e-concrete and waiting for molds to dry. But this is what it looked like before installation. So for this is just to show some pictures of the pre-installation. So the photos are showing a 0.6 foot tide on 5/31/22. And so they're just looking at the L beams here, and they're trying to see what is exposed in terms of their area of interest, and along here as well. So that was obviously by boat. Oh, and in terms of DEC permitting, so the installation that we did not have any oysters on them, so it is not a live install, so there are no seated structures. So basically the agenda of that day was that the Billion Oyster Project and Newtown Creek Alliance, with the help of volunteers and passerbys that wanted to see the installation, we got together and we utilized the 30E concrete disc and the modified oyster research station. Community members were able to view the install from the nature walk, while the team in the water and in the boats installed the structures from the boat, of course. Also during the installation, BOP and NCA, we had joined together to do Creek Critters, which is like a more informative and educational tabling event that we do that's really catered to kids, but of course to everyone. And so I was actually there able to engage the public and talk to people about what is actually found in these waters, so that was really cool. And these are just some more pictures of the installation process at Whale Creek. Just some more zoomed in photos of like in action, placing them in the L beams. And yeah, some of our crew, some of our volunteers. And that's about it.
Awesome. And then we moved on to our monitoring. So a little over a year later, if I'm remembering correctly, in April 2025, so this spring, students were able to help us actually monitor these structures. So students from the Harbor School, who are actually on Governor's Island as well with us and help us a lot in our work, they joined us at Newtown Creek at the Nature Walk, where we have these structures. And before they got started on the monitoring, they did some of the environmental history. We spoke a little bit about the Billion Oyster Project, and Newtown Creek Alliance was also able to give some history background on the area as well. And we went over the installation overview, how and why we want to monitor these oysters, if there are any right in these structures after installation, and the way we monitored is by first checking for these two different variables. So oysters might have not been what we call recruited at the time. So because we put blank structure or just blank shell in our ORSs, our oyster research stations, we wanted to also see the presence of different, so non-oyster organisms as well. So our variables were the presence of non-oyster organisms and the abundance of these animals. So first we located and retrieved our structures, which were the e-concrete discs and the oyster research stations, or ORSs. We took photos of each individual restoration structure or substrate, and then we identified them by tags. We collected our data and essentially identified the different organisms. We wanted to count those organisms as well, and then all of that data was uploaded along with the photos. So our findings were that, well, more so low abundance, because we did find these animals here. So there was a presence of amphipods, bristleworms, lacy bryozoans, and mud crabs, and ribbed mussels. So these are just pictures of the students from the New York Harbor School alongside a BOP team, the Billion Oyster Projects team, and the Newtown Creek Alliance team conducting and monitoring these structures. We did have some recommendations and just like lessons learned after this monitoring. There was a lot of sediment buildup where we had placed these structures. So as a result, we moved the structures a little closer to the mouth of Whale Creek, and we continue to develop safety and effective protocols and monitoring practices to engage more of the students and the public in the future. Next one. And these are some photos from December 1st. So this Monday, the general survey that Newtown Creek Alliance and I think the team just went out to really check on these structures and see what was out there. So here's a picture of actually a slipper snail in comparison to like this penny. You can kind of see the size of it. There's more pictures. So this is actually a ribbed mussel. You can see some smaller ribbed mussels and maybe some tunicates, some like sea squirts, I believe.
Yeah.
And this is on like the shell, so that's from the ORS's, the Oyster Research Station structures, and then this is the eco-concrete disc. I know that was a lot from like when Lewis started, so. Did I see it? Yeah.
Stupid question, but what is eco-concrete? Is it really ecological?
Kenny, do you want to speak more about the
Yeah,
materials
I
on that?
So when we say e-concrete, it's just environmentally friendlier concrete. So it's mixed with a couple of different things. So it has little remnants of shells. It's easier for critters to attach to. So the idea is that by mixing shells and like different particles, it makes it a lot easier for sessile organisms to attach to than regular smooth concrete. There are the structures at the back of the room. So like once we can get up and walk around, there's a table you can actually feel and see what that looks like.
And just to add
And
on,
that's not a, sorry.
Not an actual concrete because
It
cement is not.
It is concrete. It's just mixed with other materials. And it's not a term that the Oyster Project developed. It's, you know, a term that's used before big restaurants.
yeah, and I just wanted to mention, sessile organisms are just organisms that do not move and like are like just stuck kind of on these structures. So like the sea squirts are some of them that we saw here. And we do use this same like material, like fragmented shells from the shells that we collect in these different like molds that we use for the structures.
Did you
Yeah,
have
I'm
a
sorry if
question?
I missed this because it came late, but why are there ceramic plates on the oars system?
That's another, so like the ceramic, so in addition, so the ORS is one of the structures that we use when we're, I'll move over here, sorry, so that I'm not behind you. One of the structures that we use a lot with students. And so normally during the high season, of course, you get the things like the fish and the critters that can swim in and out of the cage, but those ceramic tiles are there so that things that are sessile, going back to what Lucian just said, students can actually analyze what's growing on the cages. And so having a surface area like a tile allows for those sessile critters to attach to. So you can analyze not only the things that are like moving within the cage, but things that want to attach to that structure as well.
I guess I kind of have two questions. One, I think you said that you didn't seed anything with oysters. Are there other places where you do, and is there a reason why you weren't allowed to? And then what is the life cycle? Like how long does it take an oyster to grow?
So I guess for this particular environment that we're working in, since it's a super fun site, we just don't have like the permitting to be able to have live oysters in these waters. But there are sites that we have across the city that, you know, would allow a live installation of baby oysters or spat into our water. And the second question was,
What's the life cycle? Like how long does it take the oyster to grow?
so I guess like the life cycle of the oysters that we've been working with are like around, I want to say 3 to 5 years. Is that correct?
Well, it depends. So an oyster can technically like live forever, but it depends on the environmental factors. So normally, like we've seen some of our oysters that we initially installed in 2016 start to reach its end of its life, and I think just because of natural factors. So that's over about 10 years for some of our projects that we initially, like some of our very first early projects that Being Oyster Project started. But I'm sure there are other places where they've probably lived much longer or shorter. So.
Yeah, I was wondering about the combined sewage overflow. Like, is it actually harmful to the oysters? It seems like they like it in maybe smaller doses, but
I mean, yeah, so I think typically we're not in favor of having sewage being dumped into the waters. There are, in certain instances, in some of the other projects, and there's a second half of the presentation we're going to get to as well about a project on mussels. We've seen things like salt marsh grasses where they've had tremendous growth. I feel like a lot of that is because, you're dumping a fertilizer into the waterway, and if you have a plant there, it's really going to grow. We see that with algal blooms as well, right? We have huge algal blooms because those are, plants that are responding to those influx. So I think with oysters in general, I mean, there's other places around the harbor where there's installations near large outfalls, and I don't know if it's
Yeah, I mean, a lot of the outfalls, there's, technically poop, but it's nutrients, right? So the oysters are filtering a lot of those nutrients, and in turn, they're growing from those nutrients. And of course, that doesn't mean, this is amazing for the oyster's health, but it does allow the oyster to feed and help to clarify the water as filter feeders. There might have been a question back here before. Yeah.
I don't know, I saw 3 hands, but.
I just wanted to ask if you were able to determine which one of these substrates seems to be working better than another, like the ORS versus the e-concrete discs or the tiles.
Yeah, I think on Newtown Creek it's a little too early to tell because this is a fairly recent installation, and so we don't have a lot of data. But I think citywide.
Yeah, I think the structures serve different purposes. So the cage itself, normally that would allow fish that are moving in and out to essentially find a home inside the cage, and you can pull that up and actually see some of the critters and crabs. But when you pull up so that's a pro to the oyster research station, the cage. Some of the cons is that, with the e-concrete disc, there's more surface area for things to attach to, but once you pull it up, everything that was either on it, whether it was a fish or crab, the mobile organisms, you won't see that biodiversity. So it allows for different types of biodiversity assessments. So there are pros and cons to both types, which is why we're choosing to do both of those structures. So that's, and it is an early project. So we just installed, oh my goodness, I'm saying it as if it was 5 years ago, but just last year. So it's still early, and I think as we continue to monitor and see what's the biodiversity looking like, we'll be able to assess, the success of both of those structures.
Yes, I got a hand. Yes.
As far as structures and placements go, are there times when you say, you have moved some of the structures out of the little inlet, or you moved them towards the mouth of the inlet because they were accumulating sediment? Is there a little more explanation of why you would cite one of the, gas station structures somewhere? And is the goal to create habitat with the structures, and how does that habitat develop after the structure?
Yeah, I mean, so with the structures that we have in, the idea is that, even throughout the harbor, one of the main issues with the New York Harbor is that there's not as much habitat for things like fish and oysters and all types of critters to attract to. And so even putting blank structures in the water increases that biodiversity from a small scale, eventually increasing biodiversity as you go up the food chain. So that's sort of one of the reasons why, I'm not sure if this was a part of your question, but one of the reasons why we wanted to install or continue to install something even without having live oysters attached to those structures.
So just to break that down a little more, baby oysters essentially need something hard to attach to, because if they don't attach to anything, essentially they could just sink and suffocate in the mud because there's nothing hard enough for them to stay up and stir and filter in a sturdy way. So that's why these structures, even if they're not seeded, it's really important because oysters can find their way onto the shell or even the e-concrete disc and actually live there and continue to filter water and hopefully not being too, that's this is another reason why we moved them, because we don't want all that sediment built up on the structures.
And there was a photo on like one of the earliest slides. You couldn't see it so well, but even on the underside of that e-concrete disc, you saw things like tube worms and like tunicates. And so life, as it builds up at a smaller level, eventually crabs and critters are picking at those little smaller animals and critters. And eventually, the more crabs and critters, the more you go up the food chain and food level, you're increasing that biodiversity by starting at the base level of having a surface for those critters to attach to. So it's literally building, you know, an ecosystem even without the presence of oysters. Yeah.
So you said that the cages were better for actually seeing like all the creatures, but the discs were better for actually getting more oysters. Why don't you just put one of the discs inside of the cages?
That's a possibility. I mean, there's always ways to re-engineer some of our structures. So we tried out a couple of different materials and structures and seeing out what works best. But I think I might take that back to our like design team. But you're on to something.
We need your name. We need your name to get the credit.
What kind of habitat would I guess originally or like historically would have been helping these oysters attach to? Would it just been rocks now it's covered with this black mayo? Like why isn't there already habitat?
Yeah, I mean, I think NCA can probably speak to this particular area, but there were lots of things. So you had a lot of tidal marshes around New York's harbor, and so with that tidal marsh and wetlands, that is a boom of different organisms. So you have things like, you know, mussels, which are still existing. Oysters were a huge part of that, which were existing. So a lot of the things that we see now were in existence, but at a higher abundance. One resource, if you haven't heard of it yet, is it's called Manhattan. And so you can look back historically at different, well, they just expanded it to include, I think, all 5 boroughs. I'm not sure if Staten Island is on there yet. Yeah, so it includes all 5 boroughs, but you can look back in time during like 1600s and seeing like what sorts of both in water and upland critters and habitats were in certain areas. So it's a pretty cool resource if you're interested in like historic data about ecosystem and different animals that were here prior to industry and destruction.
That's the website?
The new website is called Welikia.
Welikia.
Yeah. Welikia. And it's also it's a good segue. I want to take a couple more questions, but we do have another presentation about mussels, which is a good topic because it relates a lot to the ecosystems that were here. So if there's a couple more questions about the oysters, and then I want to have time for the mussels.
And then folks will be here if you
Yeah,
do
yeah.
want to like check in after.
Can you speak to any of the similarities or differences you're seeing between what you're finding at Newtown Creek and the Columbus Canal?
I would just say, well, there's no oyster restoration happening in Gowanus Canal.
Right.
And partially for the same, it's a Superfund site, so getting permits from the state is a little bit of a non-starter. It's also in Gowanus, they've started their Superfund remediation, so they've had a lot of work going on. We work really closely with groups down there, specifically Gowanus Canal Conservancy and also the Gowanus Dredgers. And it's also a good segue to our next presentation because they've been very focused on mussels, right? We've talked about how great oysters are, but there's this issue where you have to get permits from the state, and they're very timid about that because they don't want people going and eating a contaminated oyster. Pretty understandable. The mussels that are here, red mussels, are not a food source for people, so the state is much more interested in granting permission for that, and so that's something that we focused on because they're also filter feeders and they're doing a lot of the similar function. So in Gowanus, they've been looking at rib mussels as have we, and so there's a lot of overlap on that. That helps, yeah. And maybe a good segue here to Gus, who's going to run us through a project we did this year, focused specifically on rib mussels.
Cool, thank you, Willis. Yeah, by all means, we can ask more oyster questions, but we're going to pivot to another filter feeder. So I'm Gus again. I work at Newtown Creek Alliance. And today I'm talking about a project that we did this year with Phelps Dodge. So this is one of the historical industries along Newtown Creek, the Phelps Dodge Refining Corporation. They approached us, they own a property in Maspeth, the shoreline right here, and they were interested in doing a restoration project. And we really wanted to work more with ribbed mussels, so we proposed to install some ribbed mussels on their property. Willis kind of mentioned how the habitat of Newtown Creek has degraded over the last 200, 300, 500 years. And I want to look to Jamaica Bay. This is a salt marsh ecosystem. Before the Industrial Revolution, Newtown Creek was around 1,200 acres of salt marsh, and that's largely composed of salt marsh cordgrass, known as Spartina alterniflora, and then ribbed mussels. And they have a really interesting relationship. When you find them in a salt marsh, they have a mutualistic bond where the ribbed mussels are forming this little base that the roots of the Spartina can grow into. So they have a very strong physical connection. They also have a chemical relationship that we kind of found out through the course of this project. So keep this in mind. This may have been what Newtown Creek used to look like. That's changed a lot, and mussels, they're really good at adapting to these changes in ecosystem. So this was something that we put together. These are all photos from within the creek, and these are many ways that mussels have adapted to grow in niches through the creek, right? So some of these shorelines, like steel bulkheads, don't provide much habitat, but other shorelines like rocky riprap, wood, timber, any little slot or groove or crevice is actually a really great place for mussels to take root. And I'll mention something about mussels. They attach, unlike oysters, which attach with a calcium chemical bond, mussels have something called byssal threads, like literally little strings that they use to move, and they can kind of crawl across a structure, and they attach with threads onto structures. So that's why they're able to adapt like this. I'll also mention that mussels, they're super tolerant of chemicals and pollution. So when we have combined sewage overflow, not just putting nitrogen and sewage into our waters, but also things like petroleum runoff or industrial contamination, right? All this legacy contamination in the creek. Mussels are really good at not just surviving that, but also filtering it out and cleaning the water. So each mussel can clean, a healthy adult mussel can clean up to about 30 gallons of water a day. So a little bit less than oysters, but still pretty hefty for how small they are, like 1 to 3 inches. So when we were thinking about this project, our proposal was to build these wooden structures. These are made of black locust, reclaimed black locust. There's no metal fasteners in there, right? So when it's submerged in salt water, it's not going to rust. And it has all of these nice textured surfaces, angled grooves, little niches that mussels can find their way into, right? This provides secure footing. And so if you take this and you install it onto a smooth, maybe like a smooth pier piling, it creates a lot more surface area for mussels to take root. So we wanted to build about 32 of these, and then we wanted to jumpstart the rate at which mussels colonize them by actually transplanting mussels from the creek onto these structures before we installed them, right? So we wanted to go to healthy, established populations, particularly at shorelines which were due for renovation. So if a property owner wants to rebuild their shoreline, they could just kind of destroy the whole thing and destroy a whole healthy mussel population. So we wanted to go in, take some of those mussels out in advance and transplant them to our structures. And then also, mussels, a lot of people are looking to mussels for their amazing ability to withstand pollution and clean water. So we just want to get more familiar with handling mussels and working with them, because it's something that people all across the city and across the North Atlantic are interested in learning more about. So those were some of our goals. And I'm going to show you just some really great photos and videos. Here's our collection process. That's a nice little time lapse. So I mentioned that they have threads. We're actually going through. We learned that the best way to avoid stressing out the mussel is to cut their little thread. They don't feel that. It doesn't hurt them at all. So we cut their threads to pull them off the wall, put them in our little seafood boil bucket here, and then we transplanted them onto the structures. And while they were adapting, while they were reattaching to the structures, we gave them a few weeks, and we put them in this really big tank on the back of our building where we were pumping water up from the creek, filtering it through the mussels, and then discharging it back out, right? So they're filter feeders. They need fresh water to filter, carrying things like phytoplankton and other little particulate matter for them to eat. So we had to keep them fed. So we did it in 2 rounds. We kept them in for a couple weeks each time. And then we were really fortunate in that we also were doing some salt marsh restoration projects. We replanted a large section of marsh just over here. And because we had all this salt marsh grass on hand, we decided that we could kind of combine some of these interests. So we used the mussel water to water the grasses, which is really nice. The grasses want salt water, so we had the mussels flowing out into the grass tank. And then we also heard from talking with scientists at Rutgers who are developing new aquaculture techniques. We learned that having salt marsh grass physically present in the water can give chemical cues to the mussels that signal them to attach to a surface, right? So beyond having this physical structural relationship, they also take chemical cues from the salt marsh grass. So we didn't collect any hard data on it, but anecdotally, it seemed like the second time that we had this mussel tank, when we included grasses in the tank, it seemed like they attached a little bit faster. All right, here's our install. We did it in 2 rounds, so 16 of those structures in the boat. You can see there's around 30 mussels on each structure. And then here's what they look like. So we installed, about the first half, we installed vertically. So these smooth pier pilings, because they're kind of newer, there weren't many grooves or niches for mussels. So we installed some vertically, kind of replicating something like this, right? A vertical slit, which provides a lot of habitat for mussels. But we actually found out that they did a lot better horizontally. So after we installed them, when we came back in a few months to look at the change, a lot of mussels that we had transplanted had fallen off or been displaced. A lesser rate of mussels fell off on the horizontal ones because they weren't working against gravity. So we installed them in spring this year, and we came back in August and we saw a lot of new growth, right? So those orange parts, these are ribbed mussels. They have these really beautiful bright orange lips. And all that orange stuff is new growth from this year. So some of these mussels were really healthy. Some of these structures actually had more mussels than when we put them in. So they likely were colonized by some loose spat, or there were just a lot of really small mussels that we didn't notice when we were putting them on. But yeah, they seem to do really well. Here's some more glamour shots. A lot of barnacles in there. Yeah. And so we did 32 structures total all throughout this shoreline. And we're going to continue monitoring these. So just as a quick recap, horizontal structures seem to do better. We did notice some mussel recruitment within a few months of installation. We now know that Spartina might be helping in the future to signal the mussels to attach. And that as time goes on, we're going to be able to go back and collect more data and draw more conclusions about how successful this project was. And I just want to note that, since we started this, we learned a lot about how to harvest and keep and culture mussels. So we're hoping that as we go forward with new projects, we're going to have some of this in our pocket. And we've built a lot of great relationships through this, right? Speaking with people at Rutgers, working with LaGuardia students, working with volunteers, people at Hudson River Park who helped give us insight about pumps. So all these things, it's great to just build a coalition of people who want to work with mussels. Yeah, that's all about this. So happy to take a few questions before I introduce Priscilla and we start making some art here.
Are you planning on expanding the territory under that bridge?
Not at present. Yeah, right now we're focused on just going back because we want to see the rate at which they're settled with new mussels. So we're going to be going back for the next few years just to check up on it. Yeah, not so much. Barnacles are also filter feeders, but they are crustaceans, so they're not filtering to the same extent as a mollusk, like an oyster or a mussel. But that would be cool. There are a lot of barnacles already. There are probably millions of them in the creek. It would be interesting to know what effect that has on water quality. Yeah?
How often do you monitor them after you install? Do you have like a schedule?
We installed them in 2 rounds. The first one in late spring. Our last monitoring was in August. So far we've done 3 checkups total. I imagine it'll be like quarterly going forward.
You've been talking a lot about how the feeders are affecting the health of the water, but I'm curious if there are any studies that you're following that compare the filter feeders in these very polluted places to healthier spaces and look at how the water quality is affecting the health of the organisms.
Yeah, that's a great question. I think that does kind of go back to oysters. Like we noticed in our wild oyster surveys that further areas of the creek with a higher concentration of pollutants have less oysters to no oysters. Ribbed mussels, a few years ago we did a survey of ribbed mussels in the creek, and there were tens of thousands all throughout, so they seem to be pretty hardy, and throughout the harbor too. They're really tough.
I would just add too that the EPA, as part of the Superfund investigation, they did this whole ecological risk assessment, and so they actually took a cage, put mussels in the creek to see survivability in certain areas, and then they also had reference areas in other parts of the harbor, and they also did that with other species, where they would take like an organism and then like expose it to sediment from a certain part and see like basically how many of those organisms survive. So there is some data around that. But the other thing too is that I would say is that the city of New York, DEP, has also been really interested in ribbed mussels as a remedy to sewage overflow. So obviously our main path towards dealing with sewage is to reduce the outfall. But there's been some studies, including folks at LaGuardia we work with, to study, specifically removing fecal content from the water of the ribbed mussels. And so the city's taken that up and they're looking at it as well. So both of those are happening.
I know this isn't the focus on this project, but I just wanted to ask, have you all found, like, similar with the ORS cages, have you all found like a high amount of biodiversity around the areas that you all are growing mussels and things like that, like near these areas that you all have like the wood sediment?
Yeah, I mean, I think anything with a textured surface that's in that intertidal zone will attract tunicates, barnacles, and then, it's a little harder when it's actually submerged. It's harder to see what's going on the structures, but we could imagine that it's providing surface for algae, so crabs can feed on it, fish can feed on it, and it's contributing to the ecosystem as a whole. So I would love to go during a high tide and like stick a GoPro and just kind of see what's hanging out on the structures. That would be cool.
Are you doing any water quality monitoring before and after?
Oh, yeah, there are some cool projects through the city. Like I'm thinking of Chester Zarnock at Baruch is doing studies on having floating structures with mussels to see how much it improves water quality and what they're actually filtering from the water column. For us, our only data that we collect about water quality is part of the citywide community water quality testing. So that's like a seasonal program measuring fecal concentration throughout the harbor, and we help out with 6 different sites along the creek. Did you have a question?
Yeah, I was just wondering if there are multiple species of oysters and mussels that you deal with.
Correct me if I'm wrong, I think there's one species of oyster, just the eastern oyster. And then with mussels, there are also blue mussels. I think that they're not native to this exact region.
I'm not sure, but
Blue mussels.
The blue mussel that's what you eat when you go to whatever Italian restaurant or something. So, and we have seen some of those in the creek, but not nearly. They don't grow in the same way on the shoreline. So they are here in other parts of New York Harbor. You can find blue mussels, but again, you get it back into that. They're an edible species. So those are the 2 main species. There's also like zebra mussels, which people might hear about. They're a highly sort of hazardous or whatever. Problematic invasive species, but they're only freshwater, so we don't have them in New York Harbor.
Yeah, and I mentioned some of the salt marsh. This all used to be salt marsh, and salt marshes are one of those rare natural monocultures almost, where they're really largely dominated by Spartina alterniflora and then the ribbed mussels along with that. So, yeah.
Why don't people eat them?
I have heard that they're really tough and stringy and don't taste good. But I randomly read like a blog post of someone who cooked some of them and said that they weren't that bad. So that's not a suggestion to try them, but people just haven't really eaten them. Yeah. Okay, maybe that's a great segue. I want to. Oh, sorry.
I just wanted to know how you like affixed them vertically on.
Oh.
Like, how did you do it?
Yeah, so there are two pilot holes here, and so we use this was the only stainless steel or metal connection. We use big stainless steel marine bolts to just drill them onto the pipe.
Okay.
So, yeah.
With permission from the property
Yes,
owner.
with permission. Yeah, they funded the project, so we have their permission.
And also just as again, like we talked about the permits for oysters, which are very severe, we also got permits for this. So please don't go start collecting your own mussels without going through the proper channels. A lot of people get excited in this restoration stuff. We want to make sure people are doing it legitimately. So, yeah.
Okay, and now I'd like to introduce Priscilla to talk more about mussels.
You never talk too much about mussels. Hi, as you know, I'm Priscilla Stadler, and my mission I'm going to use my notes because otherwise I'll go down too many rabbit holes. My mission as a science-intrigued, creek-obsessed artist is to learn as much as I can about the science and the history of the creek, and to make art in response to what I learn, and to share that work with community in the hopes of bringing more awareness and activism to the creek. So I want to thank the Billion Oyster Project and NCA for the amazing work that you do. I love collaborating with you guys. So once upon a shell, there was an island in the creek named Mussel Island. Oops, did I do two? Sorry. And in this 1844 map, you can actually see Mussel Island. It was pretty big. It wasn't that small. It was probably a landmass that developed over a very long period of time, probably created in large part to the interaction between mussels and cordgrass and how they are able to interact and work with sediment, basically. So 30 years later, in 1874, this map maker, this is a very strange map because north is down here. This map maker, though, does leave us with a very intriguing question. You probably can't see this if you're in the back. Sorry about that. But the question that I'm left with is, why did they spell Mussel Island M-U-S-C-L-E? Was that a typo in those days, or was that just like, I'm going to leave a joke for future generations? So that was Mussel Island. And this is in the area of the creek that's near Maspeth on the Queens side, and on the Brooklyn side, I don't know, is that Ridgewood, or is that still Greenpoint? Or?
East Williamsburg, Greenpoint.
So, unfortunately, Mussel Island was another casualty of the drive for industrial profit and development. So you might be more familiar with a map like this, where there is no Mussel Island, because in the late 1920s it was demolished so that industrial ships could turn around more easily. And it's really sad, I think. But anyway, even though Mussel Island is long gone, we still today see areas of the creek, as you've seen, that, and this is Plank Road, Maspeth side, that are just full of mussels doing their thing, the native indigenous mussels of Newtown Creek. Buchancia denissa, Atlantic root mussels. So I just wonder, are these the descendants of Mussel Island? And in a minute, I will invite you to join us in a different form of crafting the landscape of the creek, which is artistic, creative crafting. And we'll make stencil prints based on my illustrations of the local mussels. My friend Fan is here to help us. It's a very easy process, fun. You can make individual prints, and I'll also invite you to make a collaborative Mussel Island banner. So I will show you how to do that in just a second. I did want to mention a couple of upcoming things, which are, if anyone is interested in making mini comics or zines related to the creek, you don't have to be an artist or have any particular skill, but artists are also very welcome. Please let me know, because I'd love to get a zine team together. And I will be having an exhibition where the Mussel Island banner that I hope many of you will contribute to tonight will be displayed in LIC in a few months. So if you're interested, just get in touch. Or if you want to contact me for any other reason, here's my info. So yeah, let's go make mussels.
Okay, so before we go into this more dispersed fashion, everyone should go and make some art for Priscilla. I just wanted to say thank you so much to Brilliant Oyster Project, Denasia, Illusion, Jada. If you have questions, feel free to stick around and chat. Make sure you go look at our e-concrete structures and the ORSs and the mussel structure, and we're going to hang out here and talk about mussels and oysters and filter feeders. So,
and also in the aquariums here are some wildlife from the creek. So there's some fish and crabs. Hart definitely knows them very well. So check those out as well.
Yeah. Cool. Yeah. One more.